students. Today I am going to discuss about the next type of cell division, that is called meiosis. Already I discussed in previous video mitosis. So when coming to the meiosis, it is a lengthy process compared to the mitosis. See how it is look like. Meiosis. So what is meiosis? Already we discussed. So what is meiosis means? One diploid cell. So majorly this meiosis occurs in where? That is very important. It occurs in only reproductive cells. Reproductive cells. Remember, the meiosis occurs in the reproductive cells. Okay, what happened in the meiosis means here one diploid cell. See, for suppose this is one diploid cell. One diploid cell, it participates in meiosis and finally it forms the 4 haploid daughter cells. 4 haploid daughter cells. So each daughter cell having n number of chromosomes. So these 4 daughter cells are haploid. Haploid daughter cells. So this is the main process of the meiosis. See, coming to the meiosis, it is a which type of process means it is a reductional division. A reductional division. Already I told you mitosis is a equational division. So that means the equal number of chromosomes are given to their daughter cells. But here meiosis is a reductional division. That means the chromosome number is reduced the half in the daughter cells. See here, parent have 2n chromosomes and finally the 4 daughter cell having half number of the chromosomes compared to their parent cell. So here what will happen? Chromosome number is reduced. Chromosomes number is reduced in daughter cells. Remember. So how the chromosome number is reduced in daughter cells? See, actually the meiosis occurs only the reproductive cells. So what are the reproductive cells in human? See, what are they? They are male reproductive cell as well as female reproductive cell. When we observe the male, what is the male reproductive cell means? You know very well, that is called what? Sperm cell. See, this is a sperm. So sperm is a male reproductive cell. What about female? Ova. So ova is a female reproductive cell. Commonly each and every cell in human body consists of 46 number of chromosomes. Remember, for suppose here it consists of 46 number of chromosomes. Here also 46 number of chromosomes. During the reproduction time, during the reproduction time, so during the reproduction time, what will happen? The sperm cell is fused with the ovum like this. So finally to form here zygote. So this is called what? Zygote. During the reproduction time, the fertilization will occur. Fertilization means what? The male cell is fused with the female cell, results in the formation of zygote. Then the chromosomes are reaches into the zygote. Total how much here? 46 plus 46. Total 92 chromosomes will formed in the zygote. So is it correct? Is it possible? Actually human cell having how many number of chromosomes? Each cell having compulsory 46 chromosome only. But here 92 chromosomes will come. Once this happen in the zygote, the cells is able to destroy. It is becomes into destroy. So it is not possible. So this is not occurs in the zygote. For that reason, during the reproduction time, the chromosome number is reduced into half here. See, the chromosome number 46. So not like this. Remember, it will be reduced into the half. Half means how much? 23 chromosomes. Here also during the reproduction time, this chromosome number is also reduced into the half. Like this. Then how many chromosomes will present in the zygote means 23 plus 23. Total 46 chromosomes. So this is the importance of the meiosis. If the meiosis does not occurs in the cell, in every cell division the chromosome number will be double. So this may be leads to the abnormal chromosomes will forms on the cells, and the cells are going to destroy, and also their genetic variations are also increases. So it may be leads to the so many problems. For that reason, in every meiosis process, the chromosomes are reduced into the half. Then only the cell will be survival. The organism will be survival. Okay, this is about the male cell and female cell. Then how the chromosomes will reduce in this process. Okay, coming to the meiosis process. Before one cell it is involved in the meiosis, before it is going to one important process. Already I discussed in mitosis also. What is the important process? Interphase. Very important. See, the interphase is also occurs in the meiosis. When it occurs means once the cell involves in the meiosis, before meiosis, the cell is enters into the interphase. See, this is called what? Interphase. So the interphase is not a single type. It has some different phases. What are the phases means? See here. See, I am going to draw the diagrammatic view. See, this is one phase. So this phase is called M phase, remember. Then the remaining part is also divided into different phases. This is G1 phase, and this is called S phase, and this one is called G2 phase. Same like that mitosis. See, this is about interphase. When the cell involves in the meiosis, before the cell is reaches into the interphase. That is very important. So the interphase is a preparatory phase, cell preparatory phase, cell preparatory phase. Remember. So what are the phases involved in interphase means, see, from here to here. See, once the cell ready to involves in meiosis, first it enters into the interphase. In which phase? G1 phase. Already you know very well. Here protein synthesis will occurs. Protein synthesis. Then after complete the G1 phase, the cell enter into S phase. Here what will happen? DNA replication. Main function is DNA replication. After completion of the S phase, again the cell is enters into the G2 phase. Here also protein synthesis. So the cell complete these three phases, G1 phase, S phase and G2 phase. Then it enters into the M phase. What is M phase here? In this process, M phase indicates meiosis. M phase indicates what? Meiosis. So once the cell enters into the meiosis, that meiosis is not a single phase. It also having so many phases. It takes more time compared to the mitosis, and it is a lengthy process. Meiosis is a lengthy process. See what kind of changes we are noticed in the meiosis. Now I am going to start the process of the meiosis. See, this is very important, meiosis. It is a lengthy process. Dear students, try to understand this process. Okay, during the meiosis, it consists of majorly two types. Meiosis is not a single type. Remember, what are the two types? One is meiosis 1. And second one is meiosis 2. Meiosis is divided into two types. What are they? Meiosis 1 and meiosis 2. So this meiosis 1, again it divide into different phases, majorly 4 phases. One is prophase 1. Second one, metaphase 1. Third one, anaphase 1. Fourth one is called telophase 1. Okay? The meiosis 1 is consists of 4 phases. What are they? Prophase 1, metaphase 1, anaphase 1 and telophase 1. Coming to the meiosis 2, it also same. It also divide into 4 phases here. What are they? Prophase 2, metaphase 2, anaphase 2 and also telophase 2. Telophase 2, remember. So this is the classification of the meiosis 1 and meiosis 2. Remember, in between the meiosis 1 and meiosis 2, one small activity takes place. A small function takes place. Where it takes place between the meiosis 1 and meiosis 2. What is the process means? Interkinesis. Remember, the process it takes short duration, very short duration. One small process will occur between the meiosis 1 and meiosis 2. What is the name of that process? Interkinesis. So what is interkinesis means? That is no DNA replication. No DNA replication. Once the cell will complete the meiosis, it does not participate in the DNA replication. So that is the interkinesis. This interkinesis, it takes a small time. That means a small duration only. Then coming to the prophase 1. Here prophase 1 is very important. Prophase 1. See, the prophase 1, it also consists of 5 subphases. 5 subphases. Remember, what are the 5 subphases means? First one is leptotene. Next, zygotene. Next, PACHYTENE. Next, DIPLOTENE. And final one is DIAKINESIS. Remember, see, the prophase 1, it consists of majorly 5 subphases. What are they? Leptotene, zygotene, PACHYTENE, diplotene and diakinesis. In each phase, the cell modified into some changes. They occur some changes. So what kind of changes we are noticed in these 5 subphases? That means prophase 1. In the prophase 1, the cell, it consists of so many changes. So what kind of changes takes place in the prophase 1? Now I will discuss. See, prophase 1. We are going to discuss about prophase 1. So, what is the first subphase in prophase? First one, leptotene. See, what kind of changes we are noticed in the leptotene? See, now I will draw the cell diagram. See, this is cell, and inside the cell consists of nucleus like this. This is a nucleus, and also inside of the nucleus consists of majorly chromosomes are present here. See, chromosomes, pair of chromosomes like this. and also here centrioles also present. These are centrioles, and here a spherical like ball like arrangement. This is called nucleolus. See what are the parts we are taking which are required for the cell division. See, this is called what? Cell. Innermost part is called nucleus, and these two are called centrioles, and this is called nucleolus, and these are chromosomes. Remember, so these are the major parts which are present in the cell. So this kind of structure which is involves in the leptotene, here some changes will occur. What are the changes means? See, first one, the chromosomes are start to condense. They start to condense. So chromosomes start to condense. So here chromosomal condensation will start. Then after, what is the change will occurs means the nuclear membrane or nucleus. So nucleus is start to disappear. Start to disappear. Remember, it is not completely disappear. The nuclear envelope or nucleus, both are not completely disappear here. They start to disappear. Remember. So these kind of changes we are noticed in the leptotene. Okay. Now coming to the second process, second surface, that is called zygotene. So what is zygotene? What kind of changes? See, this is the cell, and inside of the cell consists of nucleus. This is called nucleus, and also in between the nucleus having chromosomes are present here. And also here important cell organelles also present. This is nucleolus, and here centrioles. See what kind of changes we are noticed in the leptotene and zygotene. What is the main change means? Here the chromosomes, they are closely to near. The chromosomes are closely to near here. With the help of some chemicals, the chromosomes are closely to near, and because of lengthwise. So lengthwise, see here, lengthwise chromosomes are closely to near. Closely to near. So this kind of arrangement is called synapsis. This kind of arrangement is called synapsis. So what is synapsis means? The lengthwise, see the both side here, these four chromatids, These are one chromatids, a full is chromosome. So see here, these two chromosomes and these two chromosomes, they are lengthwise, they have equal size and shape. They have lengthwise equal size and shape. So this kind of arrangement is called what? Synapses. Then these synapses will start during the zygotene. The chromosomes, they forms as a complex. Complex means they are closely to near and also they are look like a complex. So such kind of complex is called synaptonemal complex. This complex is called synaptonemal complex. Synaptonemal complex. So these kind of changes occurs in the zygotene. Okay, then coming to the next surface. What is the next surface? Third one, PACHYTENE. Next one is called what? PACHYTENE. See, this is the cell, and inside of the cell consists of nucleus here. Then it consists of some important cell organelles. What are they? Centrioles. Then also chromosomes. See here, chromosomes, they are very closely to near in the zygotene, and next stage the chromosomes. See, the non-sister chromosomes, they are attached to each other. They are combined to each other. So what is non-sister means? See here, these two chromosomes are called sister chromosomes. Sister chromosomes. Okay, so they are aligned on the same centromere. The chromatids, see, this part of the chromosome is called what? Chromatid. So the chromatid which is present on the similar centromere. So this is called what? Centromere. Remember, the two chromatids, this portion of the arrangements is called what? Chromatids. The two chromatids which are attached to the similar centromere, that is called what? Sister chromosomes. But here these two, see, these two chromatids we are called as a sister chromosome. Compared to this, these two, compared to these two chromosomes, these are non-sister chromatids. See, these two are different chromosomal chromatids. These are different chromosome chromatids. So this is one type of chromatids, this is another type of chromatids. So these two chromatids present on the one centromere, and these two chromatids present on the another centromere. So these two are called non-sister chromatids. Remember, see here what will happen in this means, see, the non-sister chromatids, this is a pair of chromosome, and see here, this is a non-sister chromosome. This is one chromosome chromatids, this is another chromosome chromatids. In this the non-sister chromatids, they are combined to each other. the non-sister chromatids, they are combined to each other. So non-sister chromatids, non-sister chromatids join to each other, join to each other. Remember, see, in this chromosome, these two chromatids sister and these two also sister. Compared to both chromosomes, these are non-sister chromosomes. Like that, non-sister chromatids, they are joined to each other. So in this process is called crossing over. Crossing over, are also called DNA recombination. DNA recombination. So what will happen here? One chromosome DNA is transferred to the another chromosome DNA. That means non-sister chromosomes. The two non-sister chromosomes, they are joined to each other, and this is called what? Crossing over. In this crossing over, what will happen? DNA recombination. So one chromosome DNA is transferred to the another chromosome. For suppose this chromosome DNA is transferred to this chromosome. Okay, like that, this chromosome DNA it transferred to this chromosome. Like that, the chromosomes, they exchanged the DNA. The two chromosomes, they exchange their DNA. Such process is called what? DNA recombination. Okay, after completion of the PACHYTENE, the cell reaches into another phase. So what is the another phase means? That is called DIPLOTENE. Fourth one, DIPLOTENE. See, this is the cell, and inside of the cell consists of a nucleus, and also it consists of some important cell organelles which are involved in this nucleolus and centrioles. Here also nucleolus. Then coming to the important function of the chromosomes. See here, this is called chromosomes. Then here crossing over will occur in the chromosomes. After that, what will happen here means 1 minute. Okay, see, the chromosomes, they arranged as a X shape here. They are completely arranged as a X shape. The chromosomes are look like a X shape. So this kind of X shape containing chromosomes, this arrangement which are seen in the diplotene, that process is called chiasmata. Chiasmata. So what is chiasmata means? The chromosomes, they are attached to each other. They are look like a X shape in the cell. So X shape in the nucleus. This X shaped chromosomes, this arrangement is called what? Chiasmata. So after completion of the diplotene, the cell enters into the another phase, the last phase in the phase 1. What is that? Fifth one is called diakinesis. Diakinesis. See what kind of changes we are noticed in the diakinesis. See, this is the cell, and inside of the cell consists of a nucleus, but here some changes will occur. What are the changes means? Here the nucleus and nuclear membrane is start to disappear. Remember, what is the first change? The nucleus and nuclear membrane is start to disappear. So what will happen here? Nucleus comma nuclear membrane is disappear. Remember, what kind of changes we are notice here? Nuclear membrane and nucleus is disappear in the diakinesis. Then what is the next change means? Here second change is nucleolus is also disappear. Nucleolus disappear. Then the next change is the centrioles. Here centrioles are present now. These centrioles are moved into their opposite poles like this. Here also, here. So the centrioles are moves into opposite poles. Centrioles moves into opposite poles of the cell. Remember. Then here what will happen next? Chromosomes. The chromosomes, they are attached to only the terminal region like this only. See how they arrange here. See, only edges of the chromosomes, both non-sister chromosomes, they attach it to the edges or the terminal part of the chromosome only. So here what will happen? The chromosomes, they are attached on the terminal region only. This process is called terminalization. Terminalization is occurs. Terminalization is occurs. Remember, see the chromosomes, they pull to each other. See here joined, and then after the diakinesis, the chromosomes, they are pulled to each other. Just they are attached to the terminal part of the chromatid. So this process is called what? Terminalization. So these are the changes we are occurs in the diakinesis. Are you understand this? Once you take screenshot. students. Now coming to the second phase in the meiosis one. See, phase 1 completed. Now coming to the metaphase 1. See, metaphase 1. Metaphase 1. See, this is the cell. Here completely the nuclear membrane already disappear in the diakinesis. Then no need to draw the nucleus. See what will happen here. So during the metaphase, the chromosomes they are reaches into the center of the cell like this. The chromosomes they are reaches into center of the cell. The chromosomes are reaches into center of the cell. They forms as a plate here like this plate. So this is called a metaphasic plate. Metaphasic plate. Remember. And also during this time the centrioles they start to produce the some fiber like arrangements. See, this is the centriole which is present in the centrosome. Here both sides. This is centrosome, inside is centrioles. So the centrioles they start to release the fiber like arrangement which is attached to the centromere of the chromosome. So these fibers are called spindle fibers. Spindle fibers. Remember. So what kind of change we are noticed in the metaphase? The chromosomes they are reaches into center of the cell. Then the centrioles they start to release the fiber like arrangements. These fiber like arrangements are called spindle fibers. The spindle fibers attached to the centromere of the chromosome. Already you know, in the centromere of the chromosome here one special structure is present both side of the centromere. See, this structure is called what? Kinetochore. Remember, kinetochore, where the spindle fibers are exactly attached to the centromere, means in the kinetochore. Then this is the metaphase 1. After completion of the metaphase, the cell enters into the anaphase 1. Anaphase 1. During the anaphase, see, this is the cell. During the anaphase time, the chromosomes are pulled by the spindle fibers like this. See, the sister chromosomes, remember, these are the sister chromosomes. Already I told you what is the sister and what is the non-sister. Sister chromosomes means the chromosomes which are attached on a same centromere. Remember that. So here sister chromosomes they are pulled by the spindle fibers. See, this is the centrosome. Inside centrioles. The centrioles it attached to the centromere like this. Both sides like that only. and they pull the chromosomes into their opposite poles. These are the sister chromosomes. Sister chromosomes. Okay, the sister chromosomes, they are pulled by the spindle fibers into their opposite poles. So this kind of change is noticed in the anaphase. Next, after completion of the anaphase, the cell is enters into the one more phase, that is called what? Telophase. Telophase. Telophase 1. During the telophase 1, what will happen here? The chromosomes are completely reaches into the poles. See, mostly the chromosomes, they are reaches into the opposite poles, like this. So here the centrioles, they reduce the spindle fibers here. The spindle fibers start to disappear. The spindle fibers are start to disappear. See, this is the centrosome. Inside the organelle, centriole. Then here the chromosomes are reaches into mostly the poles, and finally they reduce the spindle fibers, they are reduced. So this is called telophase. During this telophase, some important changes will occur. So what are the changes means? Again, the nucleus and nuclear membrane is reappears. See here, nucleus comma nuclear membrane reappears. Nucleus and nuclear membrane. Here also nucleus and nuclear membrane reappears, and also nucleolus is also reappear. Nucleolus is also reappear, and the chromosomes start to decondense. Chromosomes start to decondense. So this is about telophase. After completion of the telophase, the cell enters into the cytokinesis. Cytokinesis. What is cytokinesis? The division of cytoplasm. How the division of cytoplasm will occur? See here, the cell, it forms some depends into inside of the cytoplasm like this. Somewhat depends. Already here chromosomes. See, these are the chromosomes. Here also chromosomes. Here nucleolus, and also nucleus and nucleolus will form. Then this depend is continuous into the cytoplasm like this. This depend is continuous into the cytoplasm like this. It will continuous into the cytoplasm, and finally the cell is divided into two daughter cells in the meiosis 1. This is the meiosis 1, and it consists of nucleus and nuclear membrane, and also chromosomes like this, nucleolus like this. So in the meiosis 1, two daughter cells will form. Two daughter cells are formed in the meiosis 1. Remember, but meiosis is not completed, half only completed. This and all what? Only the meiosis 1. Then what are the products produced in the meiosis? These products again involves in the meiosis 2. So you have to remember one thing, meiosis 2 is exactly similar to the mitosis. So what kind of changes noticed in the meiosis 2, in the next class I will discuss. Okay student, are you clear about this topic? If you like this video, please subscribe my channel and share my channel to your friends and relatives. Okay, thank you very much.